dear listeners, dear ladies and gentlemen, we are very happy to welcome Dr. Irene Margiolaki as our guest today. Dr. Margiolaki, could you please tell more about the research that you're doing in your laboratory, as well as about the goal and the mission of your lab?
Yes. Hi, Ina. Thank you very much for the very nice invitation of Deeply, this newly founded company. We are based in Greece at the Department of Biology, University of Patras. We are doing biochemistry, and more specifically structural biochemistry. We are trying to solve protein structures of proteins which are associated with pharmaceutical importance, either ingredients of currently existing drugs, such as insulin, anti-diabetic drugs, or proteins which come from the capsids of viruses that are expected to cause epidemias in the future. We use mainly X-ray crystallography in order to study the structures. So basically here in Greece, we produce the proteins we want to study, we crystallize them, then we use X-ray laboratory diffractometers in order to get first the preliminary data, and then we visit various synchrotrons around Europe mainly to collect our high resolution data, which are required for solving at atomic resolution the protein structures. And these structural models help us either to understand how these proteins function, or they are necessary for designing new therapeutics, new pharmaceuticals.
Sure. What type of proteins are you focusing on? Are there any specific families that you're focusing on, or some focus on the disease?
Mm-hmm. So there are two main axes that our lab works on for a long time now, for more than a decade. We work, as I already mentioned, in collaboration with pharmaceutical companies in Europe and in Greece on insulin, human insulin, and insulin analogs. The latter are synthetic analogs of insulin with specific modifications which affect the duration of the anti-diabetic drugs. This is the one axis, anti-diabetic drugs and insulin. This project started when I was still in Grenoble, in France, which was the place I was trained in structural biology, and keeps going. And the second axis focuses on virus proteins. We work on specific proteins from single-stranded RNA viruses, as Dengue virus, Coxsackie virus, and the Magyaro virus, and many more. And the latter is in collaboration with an institute, a research institute in Marseille, in France, which is called AFMB. Specifically, we collaborate with the group of Bruno Canard in order to solve these protein structures. The proteins have been targeted because they have an important role in the virus life cycle. We try to solve their structures and design new pharmaceuticals which will inhibit mainly the specific proteins.
Thank you for this information. The three lines of the research that you've mentioned, what was the cause? How did you come up to select these three lines of research? How did it evolve?
Mm-hmm. First of all, in principle, in my first degree, I'm a physicist. Then I was trained in crystallography in the UK, in England. And then I was for several years based at the European Synchrotron Radiation Facility in Grenoble, which is the main synchrotron in Europe, where I entered structural biology from a specific point, which was mainly to develop a technique which is called the powder diffraction technique for studying protein structures which are very hard to crystallize. That was my initial introduction to structural biology. This method is now applied quite routinely from many teams as an alternative technique to single crystal X-ray diffraction, which is the main experimental method in structural biology. And when I returned to Greece, which is my home country, it's the Department of Biology, so we started using molecular biology techniques in order to produce our proteins. We established 2 new laboratories where we crystallize, we produce proteins, and a second one where we bombard our samples with X-rays. So if I could describe myself, I'm not a pure biologist, but I come from a physical background, method development background, and these days we try to fulfill both axes, both understand and resolve a biological problem, but also further develop the experimental methods in order to explore these quite complex, in fact, biological questions.
Sure. Being on such interdisciplinary edge, coming from the physics and essentially knowing this discipline, but also contributing to the biology, as the practice shown, it produces the most of the results, and it produces the very applied research. You mentioned several diseases that you are focusing. Could you tell on the biggest achievement in the applied science that what can be also of interest to the patients as they might be watching us at the moment?
It's not only our team, but many teams worldwide trying to improve pharmaceuticals. One axis is to improve the drug delivery, and that's why we are working on different formulations of the currently known drugs, for example, insulin drugs, microcrystalline, suspensions, inhalers. We are trying to avoid injecting diabetics, for example. And the main focus of this research is to improve the pharmacokinetics of the currently existing drugs in order, for example, to reduce the number of doses a diabetic requires daily. And this way, the quality of life of a patient is, of course, improved. That's one axis, the anti-diabetic drugs. The other field of structural biology research that we collaborate with the IFMB is to design new drugs, for example, inhibitors, small organics, which will inhibit the function of the protein that we're trying to resolve structurally. And this, the small organic, in many cases, is the new drug. The same approach was followed recently for the coronavirus pandemic, and with methods such as crystallography and fragment screening, which is the new trend in structural biology, many groups have identified new small organic molecules, which will be potentially very soon out in the market, the new antiviral drugs against the coronavirus. Along the same lines, we are trying to inhibit the infection of a virus to a human organism or its multiplication, which follows the infection. By identifying new inhibitors is one way to go. And many more, of course, details that are hard to explain during this video, but this is the main philosophy.
Sure. Talking about the structure of your research group and about the backgrounds which are necessary, as well as the lab culture and how the projects are offered. Could you tell more here to provide us some insights, just to understand which kind of background is required in your biology lab to run? It can be from different disciplines, as I understand, as well as what is the lab culture, what one can expect if one joins your research laboratory.
Yes. So, as structural biochemistry is an interdisciplinary research field, usually we have many biologists in the team, but also physicists, chemists, and in some cases we had material scientists. Because, as I mentioned already, we work not only on resolving a protein structure, but also on further developing experimental techniques, which is a more familiar subject to a physicist, let's say. A part of our work is molecular biology, so it's more appropriate for a biologist or, in some cases, a chemist. Another part is pure biochemistry, where again biologists and chemists work on. Another part is the so-called biophysical part. And in that huge field, many different expertise can be facilitated. Other people work on improving or introducing new databases, which is a more bioinformatics part of our work. Other people on performing challenging synchrotron experiments, and in that aspect, everybody, all team members participate. And other people work every day in the lab locally in order to produce proteins and peptides. And this is why our team has been always interdisciplinary without barriers. In any case, I never thought that science should have barriers.
Sure, it sounds like you have a big research laboratory. In terms of the material science, talking more about these projects, what are the major axes here of research?
In terms of material scientists and why they have interest in joining our team,
yes, material science aspect of your research. As I understand, those who are watching us at the moment, they can be biologists, most of them, but material science is a little bit different discipline, and this what makes it a bit different. Therefore, people may have such interest as well. What one can expect in terms of projects in the structural biochemistry lab with material science background?
Yeah. Material scientists usually have a background in solid state physics and crystallography. So they come to us because they think that the biophysical axis is a nice extension after their first degree in material science. So in some cases, we collaborate with material scientists because people are trying to develop biosensors, for example, or thin films or surfaces to capture protein molecules, or nanobodies for drug delivery. So there are so many aspects of material science which overlap with structural biochemistry. I just mentioned a few, but there are many. Drug delivery, especially, is something that interests a lot all natural scientists, materials or chemists or physicists, and each scientist bringing her own expertise, her own background, really has a great added value, because one expertise, one degree cannot provide all this information that we require in this multidisciplinary field. So we learn from each other.
Sure. Talking about the future and the perspective of your research, where do you want to take it from here, and what would you look for? Are there any resources that you feel are still missing and you would like them to be available? Anything else? Where do you want to bring the research in the future, as well as any resources which you find limiting but you wish to have more of them, if any?
Yes. Thank you very much for this question, first of all. Of course, as you are aware, no lab can have locally all the experimental infrastructures which are required for so ambitious projects. So we collaborate, but mainly we try to use large-scale facilities. The large-scale facilities include synchrotrons, nuclear reactors, platforms that we can apply for using them. And if our proposal is successful, we get access. One of these large-scale facilities that we are trying to convince the Greek government these days, since last February, to commit to participate is the ESRF, the European Synchrotron Radiation Facility, located in France. We are one of the last European country which does not participate, and this does not give access to the Greek scientists to all these high throughput and state-of-the-art experimental infrastructures. So really for Greece, and not only other small countries like Greece, it is fundamental for the scientists to have access to large-scale facilities in order to promote their research. Without this access, we would still be, no matter how much effort we put in, we lack these very expensive infrastructures. The European synchrotron will really open up a huge way for many, many scientific disciplines, including geology, chemistry, biology, medicine, physics, structural biology. And we really hope that during this year, we'll have a positive feedback from our government.
Sure. I hope it will happen soon. We can have at the moment three types of audiences who are watching us, and this can be the young scientists, those who are interested to join your research laboratory. This can be also collaborators as well as patients and investors. We have spoken already about the application side of your research, which is relevant to patients. What would you say to those who are interested to become a student with you or a collaborator, as well as those who are seeking good investment opportunities?
Mm-hmm. Well, for the students, I always encourage them to contact us, and then within a discussion, there are so many elements which come up which may interest them. The most important thing is research, which is a long-term marathon, is to like what you do, because it's never easy. My advice is to discuss and to read. We have a team's website. If they look on Google with my name and University of Patras, they will find it. And then contact us for more discussions regarding structural biochemistry. And for investors, we have huge collaborations with the research institutes in Europe, many of them, and as well as pharmaceutical companies. I cannot name them, obviously, for confidentiality reasons, but some of them are known, some of them are on our website. Pharma industry provides us with projects, challenging structural projects, and or challenging crystallization projects. So they want us to crystallize hard molecules. Some of them are peptides, others are proteins, and resolve their structures. And what we are aiming for is to increase the number of pharmaceutical companies that we currently collaborate with. This is very important for the team, and I believe for the pharma industry, because in pharmaceutical companies, sometimes they are not familiar with synchrotron radiation, with techniques that we have developed, and they don't have locally. So I think it's a win-win situation, really.
Sure, it is very nice to hear that you're interested to increase the collaboration with pharmaceutical industry. often pharmaceutical big pharma companies, they can be sometimes resistant to collaborate with academia, though such collaborations, they bring a lot of value. How do you currently develop the relationships with such companies? What is your workflow?
Mhm. What is the subject that we work on? I already mentioned we usually take molecules which are hard to crystallize, and we combine crystallization methods from the small organic molecule world with the biomacromolecular world, because these are two different communities. And as we are lying in the middle, we can combine methodologies, which is not the orthodox way of crystallization. Or they give us projects for structurally determination of small organics, of pharmaceutical peptides. Peptides are small proteins, really, and proteins that either they don't have the expertise within the company. For example, many companies do not have large R&D departments, and that's why they subcontract teams like ours in order to complete a part of their puzzle for manufacturing a new drug, which could be either a protein or a peptide or a small organic. So either crystallization or structured solution are the two main axes that we collaborate with the industry.
Yes, thank you so much for sharing this information. Coming to the end, would there be anything else that you would like to add, what our viewers should know besides that what we already spoken about?
I think within 15 minutes we said enough. Anybody that would like more information will be happy to be contacted and provide more information. Our website is quite rich. I enrich it every day personally. But apart from that, please contact us and we'll provide any info that is required. Thank you very much for the interview.
Thank you so much for sharing the information with us and being a guest today.